All right, so we're live. Hello, everyone. My name is Aaron Alcala. I'm on the product innovation team here at EpiCypher, and I'll be moderating today's meeting. Thank you all for coming. We're going to wait a few minutes for more attendees to join, but I'll do a quick intro to our company. So we are EpiCypher. We're an epigenetics company founded in 2012 by Drs. Mark Bedford, Orgazani, Brian Strahl, and James Bone. And we were founded in response to the growing demand for high-quality reagents and tools to study chromatin regulation and enable epigenetics-focused drug development. So we pride ourselves in being a company run by scientists for scientists, and we're here to empower your cutting-edge epigenetics research. So we've recently launched some new products for high-resolution and low-cost DNA methylation mapping, which is what we'll cover today, as you can see in Brian's slide here. Okay, so we have a lot of people that have joined already, so let's get started. Just a few housekeeping items. The webinar will be recorded and available to watch in the future. The audience will be muted for the duration of the talk, but there should be a chat window on the bottom of your window where you can leave any comments or questions, and we will do our best to answer these at the end. There might also be a links button at the bottom where we will share any relevant web pages. And this is very important, but I encourage everyone to stick around for the whole talk because we will be sharing a promo code towards the end of the presentation. So now I will introduce our speaker, Dr. Bryan Venters. So Bryan is the Senior Director of Genomic Technologies here at EpiCypher, and he leads many of the R&D programs that we have. These programs seek to equip scientists across fields with robust protocols for many different cutting-edge epigenomic mapping tech, such as cut-and-run, cut-and-tag, DNA methylation, profiling, and our upcoming product, FiberSeq. Prior to joining EpiCypher in 2019, Bryan was a faculty member at Vanderbilt, where his lab studied the mechanisms underlying hormone signaling pathways. Okay, so with that, Bryan, you can go ahead and take it away.
Okay, thank you for the introduction, Aaron, and thank you all for joining us today. Today, I'm really excited to tell you about a cool technology we've been working on to solve the challenge of genome-wide DNA methylation sequencing. And really, it comes down to two key considerations, cost and coverage. So we're going to focus on the cytosine methylation. So those of you that aren't familiar, this is the methyl group that's added to cytosine. But before I get into the background, I want to give you a 10,000-foot view of EpiCypher as a company. And our philosophy is we're a company for scientists by scientists. And what that means is that we do a ton of R&D, and we keep an eye on the up-and-coming epigenomic assays to bring the coolest tech into your hands. And so you may be aware that we've brought cut-and-run and cut-and-tag to market. I'm going to tell you today about meCUT&RUN assay, and there's more to come. And one of the things we do as well is we engage in the literature by publishing how these technologies can be applied to really interesting problems. Okay, so let's talk about epigenetics. So the Waddington model is illustrated here, which effectively shows an undifferentiated cell, an embryonic stem cell or pluripotent stem cell, can essentially become many different cell types through this differentiation mechanism. And we all know now that this is largely driven by epigenetic mechanisms, not least of which is DNA methylation. And so in the human body, it's actually pretty incredible that we have over 200 different cell types, the same genome, all derived from a single embryonic stem cell. So how does this happen? Well, there's three primary epigenetic features that drive this process of terminal differentiation. The one we're going to talk about today is modification to cytosine called 5-methylcytosine, or 5-MC for short, but also histone modifications such as acetylation play a role, histone methylation and other modifications play a role in this process as well, in addition to just nucleosome positions, which can have an impact on transcription. DNA methylation has been known about for actually coming up right on 100 years. And a quick search of DNA methylation in PubMed shows you that it's really taken off exponentially. So there's a ton of interest in DNA methylation. And so from an epigenetics point of view, marks that are involved in epigenetics are those marks that influence, establish, or maintain gene expression patterns by controlling access to the underlying gene. And when we look at 5MC, its location affects the transcription. So for example, if 5MC is at promoters, that tends to be repressive to transcription. But at other locations, such as gene bodies, that can be coincident with transcription or permissive to transcription. And so we know that DNA methylation is important therapeutically because dysregulation of epigenetic features can lead to inappropriate activation of tumor suppressor genes. So here I'm just illustrating what that looks like. We have a normal gene that's unmethylated, and then when it gets methylated, it's turned off. And so you can imagine something like a cell cycle regulatory gene that is no longer now regulating the cell cycle, and that leads to dysregulation in cancer and disease. So this provides therapeutic opportunities with inhibitors that target proteins that are involved in adding DNA methylation. So there's actually several FDA-approved drugs that all target DNA methylation. All of these target DNA methyltransferases, so the writers of DNA methylation. And so I have them listed here. But let's take a look back 100 years into the brief retrospective history of DNA methylation. So DNA methylation was actually discovered by two approaches. Actually, one was a crystalline approach that I'm not going to talk about too much, but I'm showing here a chromatograph, which took calf thymus DNA. It hydrolyzed the DNA into single bases, and then it ran it out on a column. And what you see are the 4 canonical bases, guanine, cytosine, adenine, and thymine. But then there's this little blip here just next to cytosine. And this was in 1948, and Hotchkiss called this epicytosine. And this era in which it was discovered, if you recall from undergrad genetics courses, this was the time in which the base composition of the genomes and the central dogma of molecular biology was being worked out. And of course, in the mid-50s, the structure of DNA was worked out as well. So this was really an interesting time period in molecular biology. The next couple decades saw new insights into what type of organisms had this epigenetic feature. And it turns out bacteria have 5MC and also 4MC. And then higher organisms were discovered to have this mark as well. In the 80s and the 90s, the molecular biology of how the marks were regulated, the enzymes that wrote the mark were also discovered in the 90s. And then in the 2000s was the era of microarrays and genomics. And so that enabled mapping of more precise mapping of DNA methylation. And discovery of regulators of DNA methylation showed that DNA methylation is dynamic. Initially, because it was involved in terminal differentiation, the thought was DNA methylation is static. Once it's written, that's it. But now we know that's not the case. So here I'm showing you cytosine, and this is the chemical perspective that illustrates how cytosine is a dynamic regulatory hub for chemistry with these deamination, nucleophilic attacks, and this X here is where the methyl group is added to cytosine. A molecular biology view of this are writers, so DNA methyltransferases or DNMT will add the methyl group, and then these TET enzymes, these are enzymes that will remove the methylation. So this process is dynamic and reversible at some locations. And then there's also readers. Once this mark is on the DNA, there are readers of the 5MC mark. So let's talk about some of the writers. There's really 2 main types of 5MC writing. There's de novo, which would occur in embryonic stem cells as new DNA methylation is being added during the developmental process, but also de novo would occur during the dynamic process of DNA methylation that occurs during transcription, during memory formation, and other processes. And these are, the de novo are DNA methylation are regulated by 2 related DNA methyltransferases, DNMT3A and B. But the epigenetic inheritance when cells divide during DNA replication is regulated by another DNA methyltransferase called DNMT1. And so as DNA divides, it senses that one copy of DNA has the mark, and then it adds the mark onto the other strand. Okay, so the technology I'm going to tell you about today is really thanks to the genius of biology and this reader of 5MC called MECP2. And it's really an amazing affinity reagent. And here I'm showing you the crystal structure of the methyl binding domain in green, this green ribbon and alpha helix structure. And the DNA is a short segment that's symmetrically methylated. And MECP2, the methyl binding domain, is specific to symmetrically methylated DNA. So before I talk about how we used the MECP2 methyl binding protein for mapping DNA methylation in a very cost-effective and cool way, I want to give you some context and highlight some of the problems of current approaches to 5-methylcytosine mapping. So really, when you do the DNA methylation mapping, it's a trade-off between cost, coverage, and resolution. So on the left, I have some things that you would consider, such as how much material you need, coverage, cost, resolution, and so on. And so when you look at the gold standard for DNA methylation, whole genome unbiased mapping, so whole genome bisulfite sequencing, that's what WGBS stands for. Not only do you need a lot of material, and we all know this process is destructive, it breaks the DNA in ways that you might not want, but the sequencing depth required for whole genome sequencing is quite a lot, 800 million reads to get about 30x coverage. And so that the sequencing costs are quite expensive, but you do get base resolution. And so because of that, it's been the gold standard for whole genome unbiased mapping of DNA methylation. And so because of these constraints and considerations, there have been efforts to develop more cost-effective strategies that require less sequencing depth, so 150 to 100 million reads, but they've compromised on the coverage. So if you consider all the methylated CPGs in the genome, each of these approaches, whether it's reduced representation bisulfite, which uses a restriction enzyme to cut right near the CPGs, microarrays that have limited coverage, or hybridization panels, you can see that the amount of the genome that you're surveying that's of interest is actually quite small. And you do save some on cost, sequencing costs, which is great, but it's still compromising, right? So when we thought about this and we were thinking about MECP2, we thought with its ability to recognize symmetrically methylated DNA and our experience with CUT&RUN, what if we could use this affinity reagent to enrich for DNA methylation? And that's exactly what we did. So not only do we not compromise on the coverage, we're able to pick up 80% of all methylated CPGs compared to whole genome bisulfite sequencing. You do that with an order of magnitude less sequencing, and that directly translates into a cheaper assay. Okay, so how does this work? Well, our solution was to piggyback off of our work on the CUT&RUN workflow, which is a streamlined, simple workflow. And the way it works is you immobilize intact cells onto magnetic beads or a solid support, and then you permeabilize them. And the key distinction here is rather than adding an antibody against a histone modification or transcription factor, we instead add this MECP2 reader. And so it finds all the locations where DNA is methylated, and then we add protein A, protein G, eminase, which is represented by this blue sort of Pac-Man and ellipse structure. And so the protein A, protein G will find everywhere that MECP2 is bound, and then eminase will cut out those fragments of the genome that are methylated. It'll elute to the supernatant, so you have a clean separation of noise that's back in the immobilized cell versus your signal, which is in the supernatant. And so here you have the options to simply just make a library prep as you normally would for CUT&RUN. And this gives you a low resolution CUT&RUN like enrichment of DNA methylation locations. Or if you want to get base resolution and only sequence the fragments that are methylated, you can opt to do a base conversion. So here I'm showing you a very brief workflow of how enzymatic methylation works. You essentially add a combination of enzymes, TET, BGT, and ApiBac, and it essentially works just like bisulfite, where unmethylated cytosines are converted based on the activity of these enzymes and PCR into thymine, whereas methylated cytosines are unmodified. And so that works just like bisulfite, the bisulfite conversion, but it's more sensitive and easier to do. Okay, so let's talk about option one. One of the first things we did was test the sensitivity of meCUT&RUN. And you can see titrating from 250,000 cells all the way down to 4,000, locations of enrichment by meCUT&RUN effectively are unchanged. So this is a really sensitive assay. So that's really cool. And what that means is that if you have material that's very precious, or if you're working with mouse primary cells or human primary cells, and you don't have a lot, but you care about DNA methylation, this is an assay that can work for you. We also wanted to compare this with a similar technology called MEDIP. So MEDIP stands for methyl DNA immunoprecipitation. So the way MEDIP works is, rather than using intact cells and nuclei, it's more similar to CHIP in that you isolate the DNA, you sonicate it, and then you add an antibody against DNA methylation. But the problem with MEDIP is antibodies against modified DNA aren't particularly good, and it requires single-strand DNA. We compared it to MEDIP, and what we found was not only with this bar graph here, what I'm showing you is the fractions of reads and peaks, and not only is the signal to noise better for meCUT&RUN compared to MEDIP, but you can see that the signal to noise is better with less coverage, less reads. You see overall, this is a genome browser track where I'm showing you a location where there's DNA methylation represented by these peaks of meCUT&RUN, and in large part they agree, but you see that there's more noise and more spikiness in the MEDIP data. But really, the gold standard, the most stringent comparison would be comparing meCUT&RUN to the whole genome DNA methylation sequencing. So we performed whole genome enzymatic conversion, and I'm showing you a location with that track with 300 million reads, so very deep sequencing. And the red indicates locations where the individual cytosines are methylated. So this is base resolution. And when you do meCUT&RUN, you see all those locations where there's methylation with the gold standard whole genome approach. You see meCUT&RUN picking up those locations. So this is really exciting. Yeah, so there's a strong correspondence to whole genome bisulfite and EM. And what I'm showing you here is enzymatic methylation. And to get more quantitative about this, okay, it matches, and you can show one location. But if you look at every single base that's modified in the whole genome EM-seq library, how many of those are actually picked up with meCUT&RUN EM? And so what we did was we sequenced out to 50 million reads, and then we asked, okay, in the whole genome EM-seq data set that was much deeper sequenced, what percentage of those 5MCs are picked up? And you can see that we pick up increasing DNA methylation locations, and it saturates at about 20 to 30 million reads. So what that means is that you can sequence, when you're enriching for just the regions of the genome that are methylated, you can sequence 10 times less and recover 80% of the methylated CpGs. So this is really cool. So the other thing we wanted to look at was the coverage. One of the problems with these other approaches, such as RBS or MEDIP, is coverage bias. And so here I'm showing you a bar graph looking at different regions of interest with respect to DNA methylation. CpG islands are one of those regions, promoters, gene bodies, and so on. And what you see is that RBS is biased towards CpG islands and transcription start sites, where it's really not doing a good job picking up methylation, 5MC locations and these other locations, such as gene bodies, enhancers, and repeats. Whereas the blue bars show that meCUT&RUN doesn't have that bias. And when you look at this at an individual locus view, you can see our gold standard track at the bottom. We have patches of DNA methylation. And then the RBS track just above it. Not only is it very sparse, but it does a poor job at picking up the signal. Whereas meCUT&RUN has little compromise in this regard. You're picking up the locations of DNA methylation, no problem, at multiple locations with the sequencing depths that I have shown here. So I want to take a deeper dive on CpG islands, because these are regions that are often looked at in denomethylation studies. And so just a brief background about CpG islands and how it fits into the human genome. The human genome is AT-rich, which means that about 41% of the genome is GC. And so there's patches of CpG islands, and these patches are characterized by regions of high CpG density. On average, they're just shy of a kilobase in length. And they're interesting because remember what I showed in the background of tumor suppressor genes, their activity being regulated by methylation of the promoters. Many of these CpG islands overlap with promoters. And it's actually known that in cancer, changes to gene expression are often due to denomethylation changes rather than mutational changes. Okay. And so what I'm showing you here is a heat map where each row represents an individual CpG island. So I'm showing you 28,000 individual CpG islands, and the meCP2 cut-and-run signal is shown here in red. And you can see that this first cluster has a load of meCP2 signal. It may be hard to see on your screen, but there is some meCP2 signal down here. And we aligned K4 trimethylation, and K4 trimethylation is a histone modification that's a mark of transcriptionally active promoters. So if you see denomethylation at a CpG island, you wouldn't expect a nearby gene to have K4 tri, and that's exactly what we see. And then inversely, when there's K4 trimethylation, you see very little meCP2 in meCUT&RUN signal. And the three columns I have on the right are columns that give additional context to these CpG islands. The first column shows you where the transcription start sites are located. You see some are in here, but most of them are where K4 trimethylation is enriched. This shows you the CpG density within each CpG island. And then the gold standard track is shown here in this last column. And so where we see most of the meCP2 cut-and-run EM signal is where the whole genome is telling you it's located. And then where there's sparse 5MC signal, we see sparse or weak meCUT&RUN signal. Okay. So another way to look at this is to, rather than look at just CpG islands, we divided the genome into non-overlapping 200 base pair bends. So that gave us about 16 million bends. And we simply asked, compared to whole genome EM-seq that was deeply sequenced, how does meCP2 perform? So this is similar to that saturation plot, but with a flow diagram. So we have 16 million bends, and you see that most of these 200 base pair bends colored in blue don't have a single methylated CpG. And when you do whole genome, this is where you're sequencing stuff that you don't care about. It's not methylated. And so out of the 3.5 million bends that have at least one 5MC methylation, we overlap that with meCUT&RUN data, and you can see that it broke out into about 2 million of those bends had meCP2. And the bends that, that overlapped with meCUT&RUN, they were high density. And so the bends that did not overlap with the meCUT&RUN data were sparse denomethylation. And then this last little sliver of the flow diagram shows you here that the meCUT&RUN assay has very few false positives. And so what that means is 80% of all 5MCs are captured with 40 times less sequencing depth. And that translates into savings to you. So normally you'd spend several $1,000s on a whole genome sequencing study, but with meCUT&RUN, you only need a fraction of the cost without compromising on the insights. Okay, so just to summarize and to highlight the key takeaways, meCUT&RUN is a new assay for denomethylation mapping with reduced cost, low input requirements. It has better coverage than similar assays, and it only requires orders of magnitude less sequencing depth compared to whole genome approaches. And lastly, denomethylation mapping and analysis are more accessible with minimal compromise. Okay. So before I end, and thank you all for your attention, I want to highlight, as promised, the meCUT&RUN assay kit. It's on discount right now, so I encourage you to check that out. And then I want to also highlight a related assay where if you wanted to look at the overlap of the direct relationship between denomethylation and any protein associated or a genome associated protein, such as transcription factors or histone modifications, you can do that as well by swapping out the MECP2 affinity reagent with the antibody, and that's called our multi-omic cut-and-run assays. And then lastly, this fall, we have a conference in Cancun. So if you're interested in learning about the latest epigenomic insights from leaders in the field, check it out. And I've got a QR code here. And lastly, I want to thank EpiCypher, all of our colleagues that make working on these technologies and bringing them to you really fun and exciting. So with that, thank you for your attention. And I'll hand it back over to Aaron and take any of your questions.
All right. Thank you so much, Brian. That was a great talk. And we have a lot of excellent questions in the chat, and I'll go ahead and get started with those. Okay. So the first question, Brian, could you comment, we saw the data on coverage across the genome of methylated CpGs. It looked like it was 80%. Could you comment on, what's missed in that 20% and any more information about that?
Yeah, that's a great question. I'll leave it on one of these slides here. So the last 20%, that was one of the things we wanted to get out with the flow diagram. And the key takeaway was what was missed were those really sparse regions of denomethylation. And what we know about the biology of denomethylation is it's one of those marks similar to histone modifications that have broad domains like K27-trimethyl. It's rare that you find H3K27-trimethyl at a single nucleosome and not adjacent. And so by analogy with denomethylation, when it has a physiological impact, it tends to happen in a domain. So why would you detect denomethylation at one base over the span of 200 base pairs? Well, this could reflect heterogeneity in the sample. It could reflect a false positive in the whole genome. But I think the really interesting thing is most of what we know about the impact of denomethylation is captured in that 80%. And of course, if you want, you want to dig deeper. I showed you in that that saturation curve. You could sequence deeper and get to that last 20% if that's an area that you're interested in. Yeah.
Right. Okay. Next question. Will the method work with FFPE tissues?
So we haven't done that. We have an active program on FFPE. As you all know, FFPE material is much harder to work with because the heavy cross-linking, the storage conditions, it degrades the DNA. We have not applied this. I think in principle it could. Right now, we don't have a protocol optimized for that. Yeah, it's a great question. So yeah, thank you for that.
Yeah. Okay. Let's see. Another question here. How do you distinguish a region with no methylation and a region you cannot map, access, or extract with this method?
Yeah, so let me talk about extraction first. So one of the advantages of working in the cut-and-run workflow is that this is an in situ assay, so there's not an issue of extraction. With CHIP, you cross-link the material, and then you have an insoluble chromatin pellet, and then you resolubilize it. And so there used to be the view that certain regions of the genome were not extractable, so you needed an input control. That's not the case for these in situ assays. Everything is soluble in the native in situ assay. And then in terms of mappability, everything I showed you today was aligned to the HT38 build of the human genome. And so there are regions that are known to be, there's about 8% of the genome that's known not to be mappable. They're typically paracentromeric regions or satellite repeats, and that would apply to this study as well. Any short read format would be limited by the constraints of the HT38 build.
Okay, next question. Could this method work for single cell?
Yes, so again, we have not tested that internally. We do know of single cell cut and run based assays. There's a publication by Alex van Outenaarden from the Netherlands. And the way he approached it was he took single cells and a cell dispenser and put them into individual wells, performed cut and run in those individual microtiter plates. And if you can do cut and run in a microtiter plate, it's simply a matter of swapping out the key affinity reagent, so antibody for the 5MC reader. So it's possible, but we haven't done it ourselves.
Okay, this is a simpler question that we may have overlooked, but what kind of cells were the experiments from? And maybe you can comment on other cells that the assay could work in.
Yeah, thank you for that question. And my apologies for not mentioning that. All of the data that I showed you was in human K562 cells, which is a chronic myeloid leukemia line. And as far as other cells that it's compatible with, anything you can do cut and run with is compatible with this workflow.
Awesome. Okay, let's see here. We got a lot of good questions. I'm just trying to sort through them. You did touch a little bit on the human genome, but how about for non-well-established model organisms that only have a reference genome that's assembled at the contig level? How will alignment be more limited with fewer sites when using this, compared to like whole genome or RBS, for example?
Yeah, that's a great question, and I love it because it's a pioneering question, and it's a really innovative. It's a question from someone who's wanting to innovate and working with really challenging organisms. So what I would say is similar to what I mentioned about the genome build alignment constraints. The same would apply. So you can do the assay, like I said, in anything that you can do cut and run in. But in terms of the genome build, if you have contigs, there's mapping algorithms. BWA and Bowtie are the two most common ones. We use Bowtie ourselves, and there's a configuration routine that will allow you to compile the reference build. And so if you have the contigs and you're doing this custom, you can feed it into Bowtie, and then that's the space that you can view. And you'll have to tell me how much of the genome that actually represents. One thing I'd suggest is if it's a genome that you think is particularly difficult to assemble, long-read sequencing is a great way. I would check out some of the long-read publications on your organism of interest, because that long-read sequencing really excels at de novo assembly of those really challenging repetitive regions.
Right. Yeah, thank you, Brian. I actually, during my PhD, I worked on non-model, so I understand all of those problems, cross-species comparisons. And so it's great to, know that this could work for, non-well-established genomes.
Mm-hmm.
Let's see here. Here's a short question. Does MECP2 recognize DNA methylation in specific motifs?
Yeah, so this is work that was not done by us. I will touch on some of the in vitro assays that we've performed. But the MECP2 is a protein that it binds DNA methylation, as I mentioned, of course, but it's actually mutated in Rett syndrome, which is a neurological disorder. And so there's a ton of clinical interest in the protein from a neurological perspective and therapeutic perspective. And so there's been a lot of effort to biochemically characterize MECP2. And as far as impact on adjacent specificity, there's some of the early biochemical data suggested that maybe ATs and AT-rich sequences affected MECP2 binding, but then later publications showed that there's really very little adjacent sequence impact on MECP2's ability to bind DNA. And then EpiCypher, what we did was we synthesized nucleosomes that had DNA methylation in the flanking DNA regions. And then we tested the ability of MECP2 to bind with symmetrically methylated DNA, hemimethylated, and then unmethylated. And we saw no impact on the ability of MECP2 to bind. And then one of the things you can do if there's a particular sequence you're interested in, you can take all the CPG locations and plot it like a motif consensus plot. And people have done that, and there doesn't seem to be a strong bias like there is for TN5 or other enzymes that interact with DNA.
Awesome. Okay, so we may have touched a little bit on this throughout the talk and maybe in some of your answers already, but can the assay map methylation in areas that are not CPG dense? And is the MECP2 good at enriching 5MC under any circumstances, such as surrounding sequence or anything like that?
What was the first question?
Mapping methylation in areas that are not CPG dense.
Yeah, so I think that's one of the limitations, as I showed in that flow diagram, where there's regions that are not CPG dense, and the way I would define CPG dense is less than one or less 5MC per 200 base pairs. It struggles a little bit to enrich for that. And that, like I was saying, that could be a consequence of the sequencing depth. You could get that if you sequenced a little bit deeper, because we continue to pick up more.
Mm-hmm. All right, next question. Why did we choose CUT&RUN with the nuclease instead of TN5, like for cut and tag?
Yeah, that's a great question. A lot of people love cut and tag.
Yep.
We like it as well. And what I'll say is it really comes down to the complexity of the library prep. So cut and tag, to be sure, has some distinct advantages. In the context of option 2, where you do the base conversion, in cut and tag, because you're adding the adapters to the genomic DNA, those need to be specially prepared so that they're not, the cytosines are not converted to thymine, because that would impact the ability of the Illumina machines to sequence that. So that's the primary reason. The workflow is more complex to do it that way. There is a way to do it, but we opted not to go that approach because it's more complex and not quite as robust.
Okay, next. MECP2 seems to bind 5MC and 5HMC from the literature. Is there any part of this kit that can actually differentiate between those different ones?
Yeah, so MECP2, let me first say that 5MC and 5HMC, 5HMC I didn't talk about a lot in my talk, but it stands for 5-hydroxymethylation, and that TET enzyme will convert 5MC to 5-hydroxymethyl. Most of the cells, you remember that slide of all those different cell types, most cells, as far as we know from whole genome sequencing and being able to distinguish 5MC from 5HMC, most have very little or undetectable amounts of 5-hydroxymethyl cytosine. And in fact, the K562 cells that I showed you today have very little, if any, 5HMC. However, the cells that do have a lot of 5HMC are ES cells, embryonic stem cells, and neuronal cells. Now, as far as MECP2 is concerned, the literature and the biochemical characterization shows that MECP2 is specific to 5MC. There are other proteins that MECP2 is a part of a broader methyl binding domain family, and there are other proteins in that family that bind more strongly to 5HMC.
Got it.
But I should say MECP2 does interact weakly with 5HMC. It's not none, but a strong preference for 5MC.
Right. Okay. Have we ever tested intact extracted genomic DNA with a kit? Oh. Yeah, that's the question.
So more similar to the MEDIP workflow instead of immobilizing intact cells or nuclei. We've not tested that. I think that could work. I don't know why. I don't know. I'm trying to think of the advantages of doing it that way, because it's much more simple just to take the cells from culture and put them on beads and let MNase do the work. We've not done it that way. I think there is a way that could be done. It's an interesting question, but we've not done that, no.
Right. This is a This is a good question. So there are two options to the workflow. It's modular.
Mhm.
Is there one that you would recommend as the best, or how to decide between those two?
Yeah, I think it comes down to your considerations, and the considerations I think would be driven by the biological question. And so if your biological question requires base resolution or high resolution of where the 5-MCs are. So for example, if you're interested in a transcription factor whose binding is affected by methylation of a CpG within its corresponding DNA motif, then I think you would probably want to do the option 2, the conversion, the high resolution conversion. But if you're really if you're interested in the locations but not the details of where within the give or take 100 base pair window that the denamethylation is located, I think option 1 is a great option for a lot of people, especially if you're looking at differential effects. So 2 different treatment conditions, drug treatment, a mutation, a knockdown, anything like that. Option 1 is a great way for pathfinding to see whether there's an impact on denamethylation. And then you could, if you want to sequence deeper, or if you want to do the conversion for high resolution, you can. And I think that's one of the great things about, I think, this approach is it gives the agency to you to decide which level of specificity and cost effectiveness is right for your study.
Mm-hmm. Mm-hmm. Okay, we still have a few more questions here, so stick around. The next one is, can you utilize these in population studies, such as for biospecimens like buffy coat or saliva samples?
Yeah, so I think we've not done those experiments, but so if you think back to that workflow where you're mobilizing cells, so long as you can get a monodisperse suspension of cells, I think you that would be compatible. That's really the key requirement for getting the material mobilized onto the beads. If you have cell clumps, then that's refractory to diffusion of the reagents and the workflow of CUT&RUN and meCUT&RUN by extension. But so if you can get a good preparation of cells under the scope, then nice and evenly dispersed, then I think it'll work well for you.
Mm-hmm. Right. Okay. So this question's interesting. It's about, as we know, these long-read sequencing platforms, PacBio, ONT, they have a way to measure the native methylation. Mm-hmm. Have we compared our results with this approach compared to those long-read technologies?
Yeah, I love that question. So we are very interested in long-read sequencing and its ability not only to map denamethylation, but also to map open chromatin and other epigenetic features. We have not directly done a head-to-head, quite like I showed today. It's something we're very interested in looking into. It's a great question, and I think it's long-read sequencing. It's one of the things that excels at is capturing natively methylated DNA while giving you the genetics and many other things as well. So stay tuned for that. It's something we're working on. But we've not made those comparisons. I would expect that they would be similar because we compared our meCUT&RUN EM conversion to both bisulfite and to deeply sequenced whole genome enzymatic methylation, and they were extremely similar, as you saw on those slides I showed.
Right. Right. Okay. We touched a little bit on this, but maybe you can cover it for anyone that's missed it. How are you calling peaks, the software, the thresholds? Do you need an input or IgG background? And then just to follow up on that, so H3K27 trimethylation is often highly correlated with 5-MC, and those peaks are very broad and called very differently than a narrow peak like H3K4 trimethyl, for example.
Yeah. I think the answer is kind of it's a great question and a bit more on the technical side. And I would say the question is partially answered in the question, which is there can be narrow peaks, there could be broad peaks, and each one requires additional consideration. I'll say for us, calling peaks, we use the default max2, which is, we know it does a much better job of calling narrow peaks. And there's always, peak calling is a whole other subject we can get into that I don't think we have the time to get into. One of the things we did as well that I think might help is to sidestep the whole, which is the best way to call peaks, we agnostically bin the genome into 200 base pair fragments. So that's actually an approach you can do, and it works for differential methylation as well. So rather than saying this is where it's enriched, just sidestep peak calling altogether and bin the genome, and then compare those same bins.
Mm-hmm. Mm-hmm. Okay, we have a few more questions that are related to what's previously asked, but I think rewording the question is actually good to get a better answer from you, Bryan. So the next one is the 20% of the methylated DNA not covered, is it related to specific areas? In other words, is it reproducible in coverage loss or related to a variance of coverage?
Oh yeah, that's a great question. We didn't look at that. I see your, yeah. So that last 20%, we didn't look at whether it was the same 20% or whether it was sort of different regions within a collection. So I don't have a good answer to that. But what we do see is consistently when we perform meCUT&RUN, it's picking up the vast majority of the dense denamethylation regions.
Okay, I'm going to scan through because we got a lot of questions, make sure I didn't miss anything because we still have a few minutes left. So give me a second here. Okay, I think that's it. I don't see anything else in the chat. Let's see. Yeah, if we didn't get to your question or if anything else comes up, feel free to reach out to us. Check out our new website at EpiCypher.com. Thank you so much, Bryan, for leading this discussion. The other thing to note, we should have shared the promotional code for these kits, so you can go ahead and get that started in your lab. And lastly, we'll be very excited to see everyone at the EpiCypher conference in Cancun. If you haven't registered yet, rooms are limited. So go ahead and check that out on our website, which is Bryan is showing right here on the slide. So thank you, everyone, for coming. And yeah, that's it.
Thanks, everyone.